Thank you, Dr. Kaur, for so beautifully summarizing the work being done with your. And now we have Dr. Wendy Gould joining us for this Q&A session. Now, the first question I think many people may have is mini brains. What a concept, what a word. This may be a new concept to some of our community members. Can you please tell us a little bit more about this mini brain model system and describe the types of unique questions that can be asked using a mini brain model?
Yeah, sure. Now, mini brain, as we call them, and they're also called a brain in a dish, is not a human brain at all. It represents some features of the human brain. It has some neuronal cells, and it has some glial cells, and it has some mini brains are able to actually show different regions of the brain. But I just want to be very clear here that it's by no means a human brain, and it doesn't have the full architecture or the cell types that can recapitulate that of a human brain. But saying that, it's very useful in the laboratory, because with these few cell types that we can include into these mini brains, we can see some functions of the brain that can act as a surrogate for a human brain. And so we can ask questions such as, how do neurons connect with each other in this 3D model rather than a two-dimensional model, which normally we use in the laboratories to look for novel therapies and identify disease pathophysiology. These 3D structures allow us to understand how neurons connect, how they speak to each other in this 3D form, which represents that of a brain.
That's fascinating. And sort of along that line of talking about mini brains and organoids, we have a question in the chat asking, how do you envision making the step from this organoid model towards patients?
That's a very good question. So we're using these mini brains as a surrogate for a human brain, and as a surrogate and we're using the surrogacy to test novel therapies. So what we aiming to do, and this is what Simran was explaining in her talk, is to look at the electrophysiological activity in these mini brains that harbor mutations from patients, and then use that to infer whether drugs will be useful for certain patients with the same mutation. So we read these the electrical activity, so the EEGs in the mini brains, and we test drugs on them. And this will be able to tell us or inform whether those drugs will be able to ameliorate or reduce the EEG signals or the abnormal EEG signals in patients with the same mutation. So it's a very powerful approach. However, I just wanted to just highlight that it is predictive. It is a laboratory model. It is predictive, but we're hoping that this prediction may inform the use of drugs for patients.
Absolutely. And a quick question from the chat. Is this approach similar to organ on a chip?
Yes, it is. So the multi-electrode arrays are essentially like a chip, but they have their electrical platform, and the arrays are put on, the organoids are put onto these arrays, and the electricity, the conductivity between the neurons in the organoids are measured, and that is relayed as a signal, and we call this an EEG. So it's very similar, yes.
And you talked about what can be learned about specific KIP1A mutations based on the work that you're doing. I know that in Sim's presentation, she presented a list of variants that are being explored. Can you tell us, how did you select these initial KIP1A variants, and do you have plans to expand your work to investigate other KIP1A mutations?
Yes, look, the more variants that we do test, the better understanding we have of the underlying disease pathophysiology, and also treatments as well, because we know that different patients respond to different drugs. So the more we treat and test, the better. Now, the reason why we chose the ones that Sim mentioned in her talk was really to to be able to have a wide variety of phenotypes. So we selected ones that are more severe, some that are less severe, and some that are more common and some that are rare. And we thought that this would be a good approach to start off our studies, to be able to have a broad spectrum of the phenotype in which to test our drugs on.
Great. And we have a question in the chat asking, what is the time frame to identifying medications or drugs for patients? So could you help orient us to the timeline of your work and maybe let us know where you're at in the process?
Sure, putting a time on experiments in the lab is quite difficult, but I like to use short, medium, and long term. Short term, we aim to, and we are funded to test a library of drugs to be able to see which ones are effective. And we're hoping that the short term will be about 3 years. Medium term would be then to take these drugs further, to be able to test them in wider cell lines, further cell lines, maybe even, and then into a mouse model or other mammalian models as well. And then long term would be FDA approval. In Australia, it's TJ approval or EMA approval for the European community, so that these drugs can then get into clinical trials, and then from clinical trials into the clinic. Now, this can take quite a long time. However, we've started it off with a library of drugs that are already FDA approved. So when we identify drugs that are useful, as these drugs are already FDA approved, the clinical trials that will ensue from this won't be first in human. They'll be called repurposed. And so we're hoping that this will at least accelerate these drugs into the clinic because they're already FDA approved.
I love the concept of setting short, medium, and long-term goals. I think it's a great way for us to measure progress as work is being done. Taking a sort of conceptual question, the team that is working together on this grant is really a superstar team. It pulls together so many members of the KIF1A.org research network. And I was wondering if you could just share your experience in collaborating with members of the KIF1A research network, whether it's academic partners or biotech partners. How has this experience been?
All right, well, this is my opportunity to give big shout out to KIF1alpha.org. So for the community, we reached out to KIF1alpha.org. So this was Simran and I, before we started writing this grant, to ask, where are the gaps? Where are the knowledge gaps? And the powerful the power of KIF1alpha.org is that you know where the gaps are and you know who's working on everything. And so back to your question, we were able through the KIF1alpha.org to develop and put together this amazing collaborative group where we can work on this project together and work collaboratively with experts in the field, so that we can have all the experts that know everything about KIF1alpha to be able to drive this research. And we're very privileged to have met so many amazing people and to work so collaboratively together on this project. So we have scientists, we have clinicians, New Site has been absolutely amazing, and it's great to see Wayne's talk and the work that they're doing as well. So I think collectively we will be able to drive this research much faster because of the amazing resources and expertise that you have collected.
Absolutely. It highlights, if to get to the clinic, we need all hands on deck, and we need everybody working together seamlessly and in great communication with each other. And the team working on this project is such a great example of that. So we're very thankful. Another question from the chat. If the experiments show potential for improvements in a symptom like epilepsy, is it possible for that the same treatment could improve other symptoms too?
That is a very good question. Some symptoms are interconnected. Some some are not. For example, we don't know if these drugs will, if they do alleviate some of the seizure phenotype, will they then assist in alleviating or reducing some of the developmental delay, etc.? And we just don't know this. So we can, along with measuring the seizure phenotype, we are going to be measuring other phenotypes as well. We can measure only cellular phenotypes. So one thing that we can't do is measure phenotypes like developmental delay, but we can measure other cellular phenotypes as well. And this will infer whether, when reducing the seizure phenotype, that the drugs will also reduce the other phenotypes as well. And unfortunately, I cannot say yes it will or no it won't. This will be a process where we will have to investigate this while we're actually doing the studies. And then again, there's also the case that I would just want to bring up again, is that this is just inference. What we see in the lab and what we see in these brain organoids, it infers that these drugs could reduce some of the seizure phenotype and maybe could reduce some of the other phenotypes as well. But we're not too sure until we actually take these into clinical trials.
Absolutely. Another question from the chat asks, how are drugs identified as potential meds for KIF1A? And maybe we can think about this question in the context of your workflow. At what point in your workflow, if you were to identify a hit, would you say, maybe we should dig into this further?
Absolutely. So we're starting with a broad screen, and Simran and John Christodoulou in Melbourne, they've set up a high throughput screening platform in which to screen many drugs. We're not being very specific which drugs we're targeting first because, I love serendipity, and I think it's really important to screen widely. And we might find a drug that we never thought possible would actually be useful for KIF1A. And so with a broad screening, we're then going to narrow down to a few drugs, and then we're going to test these more specifically. So it's really an approach of starting broad, and then when we have identified drugs that do show a positive effect in the high throughput screening, we're then going to take these into the narrow screening. And then from there, we'll have a look at their mode of action, what other diseases, disorders they've been used for, and how they work. But we're taking a holistic approach of just screening broadly first and then narrowing down.
That's great. And, this start holistically, then narrow down really helps approach this from an unbiased perspective from the beginning, which is also very important. Another great question from the chat. Can your experiments be used to test not just drugs, I think in this context, like an ibuprofen or an omeprazole, but can it also test treatments like gene therapies?
Yes, absolutely. And that's what we are working on as well. My lab in Sydney is focused on developing gene therapies for rare neurological developmental disorders. And the aim is, in collaboration, we are able to look at gene therapies, develop gene therapies, and use these systems to test for gene therapies as well. So the beauty of having brain organoids is that when we can test gene therapies in a brain in a dish and test whether the electrical and neuron connections are restored once we've tested with gene therapy. So the systems are very it's multi-purposed, and it can be used to test many different types of therapies.
Great. And a great follow-up question from Luke in the chat. Would it be helpful for the community to survey which drugs have not worked on KIF1A seizures to eliminate and help efficiency? How would that integrate into your workflow?
So that is a very great question. So what we do with this platform is that we, along with the novel molecules that we test known drugs as well. And the data that we the data that we use from this is to see whether if drugs that are known to work on individuals, or that don't work on individuals, or where individuals have become refractory over time, if we see this in our platform, then we know that the platform is working well. So we do this currently where we, on other disorders, where we speak to the neurologists that are treating individuals. We ask them what drugs they're on, what drugs they have become refractory to, and we use those drugs and test them alongside the novel drugs to ensure that the system actually is valid. And if the drugs that the individuals have become refractory to, if they stop working over time and don't show the signals that we see in the dish, then we know that the model is valid.
Looks like we found a call to action. Thanks, Luke. That's absolutely something we can work on. Another question. If you find several promising drugs in your screening, will you be able to follow up on all of them in parallel? How do you pick which drugs to invest time and experiments in? And I'm going to add on to that. If it's not possible to address them all, what do you need and how can we get that to you?
Yeah, so that's a great question. It comes down to resources, really. So that's funding to support the resources in the laboratory and the people to do them as well. I think that's a bottom line. It comes down to resources. We have the capacity to be able to follow a number of drugs in parallel. The other thing that is also important is collaboration. So it will be very critical that if we do find a number of drugs, say we find 10 that are really great, then through collaborative efforts, we can then divide up the number of drugs that we want to test, and different laboratories can test different drugs, so that in synergy and in parallel we can drive these drugs closer to the clinic faster.
As we like to say at kefone.org, science is better together, and that's an excellent example of how working together gets us to the clinic faster. Let's see. What is the biggest challenge to creating a drug screening system like what you are building?
Time is the biggest challenge. So with a high throughput screening, we call it high throughput, but it does take a lot of time because just to remember, these are cells that we're working with. It's a biological system, and they take time to grow, they take time to screen, and they take time to analyze as well. So the biggest challenge is time. The other challenge which is really important, is reproducibility. We can do an experiment and analyze the data and pick 10 top drugs that we think have a good chance of making it through into our medium throughput analysis. If we repeat that on another day, we might find 10 different drugs. So this comes down to time as well, but we need to make sure that every time we do the experiments that we get the same or more or less the same drugs that are our top performers. And so with the work that we're doing, it not only takes time to set up the experiment, to do the experiment, but it takes time to repeat it and validate it to make sure that we have the right drugs and that they're reproducibly performing the best, so that we can then move on with those. The last thing we want to do is pick up something that is a red herring, and it was just a mistake, or maybe not a mistake. Mistake's not a good word. It was just a once-off effect, and we can't reproduce it, and we don't want that at all. What we want is something that's robust and reproducible, so that we can be certain that these drugs that we have identified will have a high chance of being effective in the clinic.
Absolutely. Time is such a challenge in so many ways around this common problem that unifies us. Another example of why many hands working on the same problem is increases efficiency, and a comment from an anonymous in the chat just wanted to say, I love that you're also working with medicines that are already FDA approved and will just be repurposed so the process will be faster for patients. That also addresses that time component. One more question. Do kyphonite organoids show a similar phenotype as wild-type organoids, or does kyphonite also impact the growth of organoids?
That's a very good question. We haven't grown the organoids yet. We're growing the neurons, but we haven't grown the organoids yet. So in our lab, we haven't been able to show a distinct phenotype. But in a lot of neurological disorders that have been published and that we're working on, they do show a phenotype. Some even show, some are even smaller, some will be bigger, some will have different features such as different number of synaptic vesicles, differences in mitochondria, and we're expecting a specific phenotype with the KIF1 alpha mutant organoids, but at the moment we haven't got to grow them yet, which is something that we're working on.
And I know you will update us when that time comes.
Absolutely.
It looks like we have time for one last question, and this is I like it. It's about a cross-disciplinary approach to therapeutic development. It says similar drug screen approaches on tumoroids are going on in the oncology field. In fact, many startups with lots of funding. Any chance you could benefit from these efforts in oncology to accelerate?
Yeah, that's a very good question. Absolutely. There's so much crossover with all the work that the different disciplines are working on in the field, and it's definitely something that we can look into. We work very closely with a team that works on neuroblastoma, and they grow brain organoids as well. And we meet with them regularly and cross-pollinate ideas and theories to be able to improve our work.
Excellent. That's all we have time for today, but I want to extend a huge thank you to Dr. Gold for joining us so early in the morning, for starting your day with us over in Australia and sharing with us your work that is really accelerating epilepsy treatment in children with CAN. We are so inspired by this collaborative team of researchers that are working on this project, and we can't wait to see how this team will advance our therapeutic development for CAN. So thank you very much. And thank you to Dr. Kaur as well for your presentation. All right, we are at our last speaker in this section of the conference. So next we are going